Okay, hello everyone. I'm Jennifer. I'm currently a PhD student at UC Berkeley, and today I'd like to share with you the method we developed, RISE, which compare the relative impact of splicing and expression in RNA sequencing studies. RNA sequencing study has been widely used to quantify expression and splicing changes in transcriptomes. Although biological consequences usually arise from both expression and splicing, researchers usually use their impression to choose one to analyze. Uh, potentially overlooking significant impacts of the other. Even if researchers try to analyze both, the measurement scales of expression and splicing is on totally different scales, and as a result is incomparable. To compare the relative impact of expression and splicing in a uniform way, we have developed RISE. RISE is not only a new method we developed, probably say that RISE is more like kind of a conceptual change that we are trying to propose. It is like defining the RPKM that for expression, it provides a formalized way for us to discuss or handle this kind of data. And with RISE, hopefully like now we are able to have a common language to talk about this in a way that we just never talked about it before. So to calculate the expression and splicing changes, we first aim to calculate the between group variation of control and treatment group label in red. The between group variation can be calculated by subtracting the within group variation from the total variation. By doing so, we remove the noises between the replicates. Within group variation is calculated in green or blue, and total variation can be calculated in black here. And we can do the same analysis for the slicing part. And till this step, you can see that the expression and splicing changes are totally on different scales. So these numbers are still incomparable. To put expression and splicing on the same scale to compare their relative impact, we calculated a term we called normalized variation. Normalized variation is defined as a proportion of between group variation over total variation. And so we calculate the express normalized variation and splicing normalized variation for each gene in the transcriptome. And this normalized variation allows us to compare the relative impact of expression and splicing. And finally, we assess whether expression or splicing has a greater impact by a t-test to determine which one has a greater effect. So with this RISE method we developed, we next sought to kind of test RISE to see if that really works as we proposed. In order to validate RISE, we perform RISE analysis on several several transition and splicing factor perturbation studies. So for example, take transition factor as an example. When the transition factor is knocked down or overexpressed, you would expect to see larger changes on expression than on splicing. And likewise, we would expect the opposite outcome for a splice factor. So this is the summary result we got. In this figure, each data point represents an RNA sequencing data set of a transition or a splice factor being perturbed. The x-axis represents the expressed normalized variation, y-axis represents the splicing normalized variation. And by having this kind of MV concept in RISE, we now are able to put them on the same graph to compare them. Before this, we wouldn't know like what kind of figure to make sense of it, or we probably wouldn't know like how to interpret it. But now with RISE, we are able to compare their relative impact. And actually, RISE also did quite well. It actually turned out that RISE can effectively categorize most of the transition factor and the splicing factors. So in this figure, you can see that all these transition factors, when they are being perturbed, RISE detect larger expression normalized variation when compared to splicing level changes. On the splicing level for these splice factors, most of them all have larger splicing level changes compared to expression level changes. However, we do also have like one unanticipated result of one of the SRF3 study. So despite SRF3 being a splice factor, RISE did not detect significant larger splice impact in one of the SRF3 study. The discrepancy was not recognized until analyzed by RISE, but the reason for that is still a little unclear for us. And in addition to validating RISE on those known transition factor and splicing factors, we also thought to try to apply RISE on some proteins with kind of complex or previous unknown roles in regulating transcriptomes. So today in my presentation, I will show you one example here. In this study, the group was originally interested in understanding the role of this RNA binding protein SELF6. So they overexpress SELF6 to want to understand their role in tumor, their potential role as a tumor suppressor in lung cancer. So the authors find several genes are change the expression and splicing changes. They found both changes on these two aspects, but they didn't know what's the relative impact of expression and splicing. And so now with RISE, we have a formalized mechanism to discuss this. And actually, before I move to the next slide to reveal the answer, I just want to maybe have a vote here. So for people here, do you think like when you overexpress SELF6 protein, do you expect to see higher expression changes or higher splicing changes? Okay, maybe for raise your hand if you think it will have a larger impact on expression changes. 1, 2. Okay, just 2. How about people who think that it will have a larger impact on splicing? Yeah, more people here because it's an RNA binding protein. I think all of us have this kind of an intuition. But actually, with RISE analysis, we see that the SELF6 overexpression actually had a greater impact on expression. And the reason behind this might be that SELF6 also have some roles in regulating the stability of mRNA. So it might regulate the stability or the metabolism of mRNA. And in order to understand more for each data set that I presented in the last slide, we can also zoom in to check the expression and splicing changes of each gene, the transcriptome. So in this density plot, like the color represents there are more genes in that area. So for example, you can see that with RISE, we can see subpopulations of genes with distinct expression and splicing level changes. For example, in the pic to your right-hand side here, these genes all have high expression changes and low splicing changes. And we label some of the genes that was also identified in the original study because of their high expression changes. So this is kind of consistent with our analysis. But in addition to that, you can also see that there are some genes here, they have high expression changes, however, also have some moderate splicing level changes. So this MYD88 gene was initially identified in the original study because of its high expression level changes. But here with RISE, we can see that it not only have high expression changes, it also moderately differentially spliced. And in addition to that, the picture left here, genes here have low expression changes and moderate splicing changes. This suggests that these genes here might have alternately spliced, but they are still kind of like expressed at the same level. So in conclusion, RISE provides a kind of new nomenclature that allows us to compare the relative impact of expression and splicing. And we hope that this can help us to kind of see things that we might previously overlook. Yeah, so with that, I'd like to thank people in my lab and appreciate their support, and always down to get boba, the drink here together. And I actually will also present another poster about variant interpretation database on Monday. If you're interested in that, I'll happy to talk to you about that. With that, I'll be happy to answer questions. Yeah.
Okay. So yeah, great talk. Of course, in this assumption that that you should be able to predict whether you get an effect of gene expression or in terms of splicing, so you sort of have built in a hypothesis of direct effects, right? Splicing factor affects splicing, transcription factor transcription levels. Of course, there can be these indirect effects, right? You could have a transcription factor that affects 6 splicing factors, which then control 5,000 exons.
Yeah.
So do you think that could explain some of the discrepancies?
Yeah, I agree. So I agree that some of these transcription factor or splicing factor definitely would have like secondary effect. That's something we're also kind of worried about. But so in the current data set, I think most of the effect we're observing are probably mainly primary effects. But I agree that if there are some like kind of discrepancy, those might be potential reasons for that. Yeah.
Thank you.
Yeah. Okay. Thank you.